Okay, so now we're going to talk about the G protein coupled receptor. Like I said, this is going to be one of those complex concepts where you'll need a paper and try to draw this out. And you'll need to know all the steps. You'll need to know all the enzyme involved and the sequence that this happens. Okay, so G protein, as we've talked about, it got its name because it's bound to GTP. So it's a protein that's bound to GTP. And then there's this receptor called G protein coupled receptor. So it's a receptor on the cell surface where it's coupled to a G protein, which understandably have GTP or GDP on there. Okay. When you use my PowerPoint, make sure to use the following slides in the presentation mode. That's how you're going to see the movement and the animation. Okay, so let's look at this. What do you think this is? Yep, that's the nucleus. And then what is this? That will be the membrane. So again, we're giving you an example of the cell surface receptor. So the receptor will be on the cell surface, and you'll need to know this GPCR as a detailed example of cell surface receptor. So here is our receptor, and it has all these helical structure, okay, and folding because it's trying to get through the phospholipid side, and that's actually how membrane protein folds in alpha helical structure. So right here is showing you the structure of a transmembrane protein. It's a receptor. It's sitting on the membrane, and it's bound to a G protein. This is a G protein. It has 3 subunits. Remember, we talked about how protein has potentially a quaternary structure where different protein come together. Well, this is one of them. There's 3 that comes together. What's their name? Alpha, beta, and gamma. And it's bound to GDP currently, thus the name G protein. Okay. Then what does it make this whole thing? Well, this thing then is called the G protein coupled receptor because it's itself a receptor and it's bound to a G protein. Okay, so how do I activate G protein coupled receptor? Remember stage 1, you need receptor activation. How do you do that? Well, we need a ligand. Okay, so step 1, this ligand needs to bind to your G protein coupled receptor, GPCR. That's step 1. Now, remember we talked about when it bound, then your receptor will change conformation. Okay, once it's bound, it's going to change conformation, this GPCR. So step 2, GPCR, change its shape. Now, as it changes its shape, then it's going to dissociate this G protein, as you will see. This G protein now is affected. So it's a chain reaction. You got ligand coming on to GPCR then affects the G protein that it's bound to. Okay, so it's like a cascade. Third, the alpha subunit right here, this is alpha, is going to exchange GDP into GTP. So it's going to become a alpha subunit with GTP right here. So as it changes to GTP, it then can move away from the receptor. So it's no longer bound to the receptor. Okay, now it's active, right? We talked about this. You go from diphosphate into triphosphate. Now you're activated, okay? And so step 4, alpha subunit GTP is going to now dissociate because it's active, and then it's going to go, okay? This alpha subunit is going to go somewhere. So what's going on right here? Once it's activated, it leaves that GPCR, okay? So that's gone. It moves away. And then this alpha subunit with GTP is going to go one way. The gamma and beta is going to go another way. Look at this. Okay. And where does this go? Well, this alpha subunit is going to go to another enzyme, ACE, called adenyl cyclase. Okay. And activate adenyl cyclase. Okay. So number 5. the alpha subunit with GTP is going to activate adenyl cyclase. And adenyl cyclase does what? Well, step 5, adenyl cyclase actually make cyclic, so round cyclic, AMP, adenosine monophosphate, from adenosine triphosphate from ATP. So now you can understand the name of adenyl cyclase because it actually makes ATP into cyclic AMP. So it's an enzyme that makes cyclic AMP, okay? So it's a cascade. One nudges the other activates or moves, and then makes the next thing happen. In this case, we're now with cyclic AMP. Number 6, once cyclic AMP is made, it then moves and binds to the next person, and that person is called protein kinase A, pKa. Now remember kinase. What does kinase do? What did we say about kinase? It's an enzyme that adds phosphate, so phosphorylates. Okay, so as cyclic AMP binds to pKa, then now pKa is activated. And this activated pKa can then move on and then phosphorylate many other response proteins. So pretty much that's all you need to memorize up to pKa. You got to know all the names before that. And up to this point, you just need to know once pKa, the kinase, is on, then it goes and phosphorylates a lot of different proteins. So this is like you finally got to the control room and you get to switch on many buttons. And now the cell is completely activated to finally get you the cellular response. So number 8, cascade of response protein finally lead to cellular response. Okay, so you can stop the video here and see if you can recount these 8 steps. Another thing you can do is after you recount these steps, then try and draw this out. Okay, so I'm gonna pause the video and try to see if you can talk through what's going on in here. And there will be answer in the next slide. So first work on this. Pause the video and work on this. Okay. Draw the pathway out on a board. So you can draw it on a piece of paper. And then you can indicate how all these works and map back to the 3 stages of cell signaling. Can you do that? So if you've drawn it out, can you tell me which steps is the first stage? Remember the 3 stages? Receptor activation, signal transduction, cellular response. Can you map the steps back to the stages? Because we are asking you to do this example exactly for that reason. Now you can see ligand binding to GPCR and conformation change. That is stage 1. Okay? Everything after that, 1 to the next, 3, 4, 5, 6, 7, that's all stage 2, signaling transduction. And now you can see it in an actual example. Last, stage 3, cellular response. That would be step 8 in this GPCR cell signaling. So to conclude, we're kind of telling you all these different cell signaling examples, okay? But then we introduce that there's 2 main types according to the ligand. So if ligand is hydrophilic, then you can't pass. Like this ligand, it can't get into the cell. So then you'll need to match what kind of receptor. Yes, cell surface receptor, okay? And then we give you 3 different examples, chemical gated ion channels, right? And the enzyme receptor. And then now the main one, GPCR. And then we tell you exactly the detail of GPCR. The second type, hydrophobic ligand. Well, it can go through the cell membrane, right? So it will have intracellular receptor. Now let's move on to that. Okay, so you can watch this video on your slide. I'm going to move on to giving you also an example of intracellular receptor. Now, the main example you can think of for intracellular receptor will be hormones. Okay, these are usually nonpolar, so they are hydrophobic, and they can cross the plasma membrane. So their receptors are usually inside the cell. Now let's talk through this. This red looking shape molecule is the hormone. And you don't see any cell surface receptor because its receptor is inside. Okay, so first step is hormone is going to cross plasma membrane and then bind to its intracellular receptor. Now look at this intracellular receptor. It looks very interesting. Okay, this darker blue is bent and it's bound to something called an inhibitor. So do you think it's activated? It's actually not activated. It's being inhibited. Now what happens? Stage 2, when hormone binds to alter the receptor conformation, right? So now do you see that it then changes shape, okay? So you got your hormone binding alter receptor conformation or shape. And what it does is as it changes, it now releases the inhibitor. So you see how conformation change, it goes from this to this, and now the receptor is, the inhibitor is gone. The hormone's still on it, but this shape change, okay, now allows this to do something it wasn't able to do before. It can now go through the nuclear pore and inside the nucleus to actually change gene transcription, okay? So now it moves in and you got receptor and hormone and receptor complex going into the nucleus. Then this hormone receptor complex can then do what? Once it's inside the nucleus, what's in the nucleus? The main thing right here, DNA, okay? So this receptor can now act as a transcription factor. It calls for RNA polymerase to then bind to certain genes and start making RNA. Okay, so it's turning on genes. So this hormone receptor complex binds to DNA and can then turn on or turn off gene transcription or gene expression. So the cellular response is a change in the gene expression. Remember, we got 3 cellular response. So usually hormone, right? All these estrogen, testosterone, they are fundamentally changing how your cell react through turning on and off genes. That's why when you have estrogen, it talks to all these cells and say, you got to proliferate more fat cells, okay? Or you got to do this other thing. And that is through one of the cellular response called changing the gene expression. Okay, so all the answers are in the next slide. And it's unpacked here. Binding of the hormone to the receptor causes the complex to shift from cytoplasm to nucleus. And now it's a transcription factor. It can make RNA. RNA can then be translated into protein. And now the cell can really change significantly. And this type of hydrophobic ligand or hormones can act as regulators of gene expression to turn genes on or turn genes off. Okay, that is it. That concludes chapter 9.